necrosis of some subset of lymphoid cells, but also of infiltration at the inflammation site. I think it's a combination of both.
Okay, so another question is related actually to the level of C5. Where would you situate or locate C5 regarding the cytokine storm, for instance? So one question was about whether we could use the C5 axis as a warning actually against the cytokine storm. And whether if you use this, the second actually linked question, if that's the case, then if you use, for instance, C5 as a target, would be no longer using or no longer needing to use an inhibitor of the cytokine storm if it's ahead, if it's earlier in the pathway.
That's a very good question. As you could see, I didn't claim that C5a was upstream of the cytokine storm. I was just saying that there are data supporting that it could be the case. But I think that it's likely, and this is my personal feeling, is that C5a is obviously not the only factor to initiate the cytokine storm, but it contributes to the cytokine storm. And it contributes to the cytokine storm in two ways. First, by inducing the migration of myeloid cells to the site of inflammation. This is obviously very important. The production of cytokines within the inflamed lung is a product of two factors: the number of cells that can produce the cytokines and the amount of cytokines produced by one given cell. So you have to multiply them to have the total explanation of the cytokine production. So for sure, C5a is a very potent chemoattractant molecule. And I've been showing you that both in vitro using neutrophils from healthy individuals, but also in the C5aR1 knockin mouse in the acute lung injury. So I do think that this is a way that C5a contributes. Now, whether C5a can be monitored in COVID-19 patients, yes, obviously. Okay, this is an important factor, and whether it's a warning factor, we still don't know that. We need many more patients to be fully convinced of that, and whether one can treat or not patients using anti-cytokines in the combination of durvalumab or other blockers of the C5a, we just don't know. The clinical trials are actually made for that. They are made to answer to the question whether the blockade of the C5a-C5aR1 axis can be of interest for the patients. And as I've been telling you 5 days ago, there are very interesting data showing monotherapy results with an anti-C5a blockers.
Great. Eric, do you think that C5 targeting would help with dexamethasone?
Okay, so that's a very good question. Yeah, again, I'm not some kind of prophet. What I'm trying to do is to translate the data from basic science into possibility of treatment for the patients through clinical trials. So as long as the clinical trials are not held, I would certainly not tell you anything on the results. But what I can tell you is that it makes a lot of sense. Now that we know that the blockade of the C5a pathway has some interesting signal in this phase 2 clinical trial from our colleagues at InflaRx, it makes sense indeed to think of possibilities of developing clinical trials in association with other molecules. And as the UK group released information for the role of corticoids in COVID-19 patients, why not this kind of molecule, obviously?
Another question is related to whether you have checked the C5a-C5aR1 axis after recovery.
I'm sorry, I don't hear you very well.
Yeah. So another question. Yeah, another question is related to whether you have checked the C5a-C5aR1 axis after the recovery of patients.
No, we didn't do that. It's a good question. I don't have the results for that, unfortunately. I don't expect to see a major difference in C5aR1. As I told you, C5aR1 is really expressed on 100% of neutrophils and circulating monocytes. it aspects as steady states is not the limiting factor. The limiting factor is C5a itself. So we don't know for the patients after recovery. All I can tell you is that C5a is not detectable in healthy individuals. So my expectation would be that if we would look at C5a and C5aR1 in patients after recovery, C5a would be down and C5aR1 would be still very high. Because again, for C5a to be able to do something, it needs to recognize, for C5aR1 to do something, it needs obviously to interact with C5a. And C5a is the limiting factor.
And you think that's when you look at the expression actually of the C5a receptor, it's almost always high, right?
Yes, it is.
Right, in contrast to C5a, which would go up,
yes.
With the severity of the disease. So that
yes.
means that's even. So how do you explain that, actually, Eric?
Well, as I told you, I mean, C5aR1 is expressed at steady state on all myeloid cells. Okay. Yeah. But it doesn't do anything unless it encounters C5a. So C5a is induced during inflammation. It can be actually induced here directly by the virus, by this N protein of coronavirus, but it also can be induced by CRP, by the way. It can be also induced by, obviously, antibodies. This is a classical pathway of complement activation. So there are different pathways that could lead to C5a activation in COVID-19 patients, and this is a limiting factor.
Yeah, another question. You let me know if you're tired, Eric.
No, I'm fine. I'm fine.
Is there an age difference in terms of the expression of C5a?
Again, I was probably not clear enough. What is the key difference for C5a is the level of inflammation. If you have inflammation, if you have infection, you can have C5a. I mean, this is well known for a lot of chronic inflammatory diseases, such as vasculitis and other inflammatory diseases. This is a case for this coronavirus infection. So we didn't look at kids in particular. Whether C5a could be also present in kids upon SARS-CoV-2 infection, if this is a question.
Okay. Another question. Another question is related to the type of macrophages which are prevalent in the COVID-19 patients. Do you have an idea?
No, so we didn't look at this in detail. Good question. Other groups have done that and are doing that. So I'm not going to say more than what I've said. We say expression of classical myeloid cell markers, CD163, CD68.
A question is related to IL-8. I'm not sure if whether an attendee asked about IL-8. What was the level of IL-8 in the bloodstream? Did you look at that?
Yeah. So we did look at that. And IL-8 was particularly very high in the broncho-lavage fluid. I've been showing you the result of that, which is extremely important because if you have IL-8 in the inflamed lung, you can have a rationale or mechanism, a potential mechanism for the infiltration of neutrophils in the inflamed lung. So yes, we did see IL-8, CXCL8.
Okay. You looked mainly at the antibodies, I mean, blocking antibody against C5aR1. I mean, whether you think targeting C5aR1 is better than targeting C5a, the soluble factor? I mean, the ligand?
No, I'm not saying that. I'm not saying that is better. Yeah. Again, I mean, only the clinical trial will tell us. The clinical trial, the phase 2 clinical trial addressing the role of C5a in COVID-19 patients has just been released. Now those colleagues are thinking of going into phase 3. We are still recruiting patients in phase 2. So once we have the result of phase 2, we will compare the results. It's definitely the same pathway. Now, whether pharmacology speaking, or the saturation of the molecule, saturation of the receptor, saturation of the ligand might have differences in terms of efficacy. I just don't know. But you mentioned also that we were using only antibodies. That's true. Although in vitro, we have been using also avacopan, which is a small molecule that blocks C5aR1. And at least in vitro, we could see that there are comparable action of C5aR1 antibodies, have C5aR1 antagonist, and anti-C5a itself. So,
yeah, actually, another, I guess, last question is related. Usually, when we talk about COVID-19, we talk about a viral stage or viral phase where we can use antiviral molecules, and then an inflammatory phase where we can use basically anti-inflammatory molecules. And although you have stressed it much here in terms of immune checkpoints, basically release the immune system to, to do its job. Where would you locate that? Is it in between both phases?
Okay, so they were very effective, which I think it can be disputed at present. As for immune checkpoints, yes, we have studied them, in particular in lymphoid cells. We have been paying attention to PD-1, to CD39, to NKG2A, and as I mentioned very briefly in the introduction, in the interest of time, I didn't go through the result, but this is in press in cellular and molecular immunology. So we've seen an increase in NKG2A, we've seen an increase in CD39, an increase in PD-1 on NK cells. We could actually document an increase of NKG2A as well on CD8 T cells. So there is an idea that one can actually unleash these immune effector cells, because we know that NK cells and CD8 T cells are so important in the control of virus